Oh, so I humility class. It's really awesome. Okay, so we're in the M chapter, metabolism, and we're talking about the body getting rid of the drug. And I kind of was thinking about this philosophically this morning, and I was like, when man kind of hit the scene, whenever that was, there was no Merck and no pharmacists, right? So here I am, and I'm eating stuff. I'm eating grass or berries or mushrooms or some kind of fruit, right? So why would we have P450 in our liver, right? Why would we have drug metabolizing in there? They're really probably not drug metabolizing. They're probably foreign substance metabolizing them, right? Drugs will into that as well, right? So if you're like a berry collector and you pick the bad berry, I never understood why you can't eat those berries, like the bird meat. Like my mom was a candy boy. I could eat them. And they used to fall on my car all the time, stain my car. Again, too much information. But I tried one once, and it was so bad. Right, so anyway, I must have been a little. So okay, so why would have this? Well, they're there. They're there, and so they do something, and they do something to these foreign substances coming into the body. Xenobiotic, foreign Greek biotic agent, foreign agent. So, yes, liver, obviously, its function is critical, because we talked about that, how there's enzymes there, oxidation reactions. And so, one of the themes of this chapter 4 is the fact that there's variation in patients, right, the drug responses, and that's really a continuation of a chapter 2 concept. Remember we talked about clinical dose effect curves. We said some people are very sensitive to low doses, and some people are very resistant and in higher doses, and then most people are kind of in an average area in the middle, normal distribution. Right? So when you look at that cohort, you can't say that there's no variation. There's variation. There are people above the mean, there are people below the mean. There's variation. Why is there variation in drug response? And so one of the things our book talks about is disease. There are variations in drug response because of disease states. Right? So you're about to embark on this tremendous journey next year, the drugs and disease courses. Right? The drugs and disease courses, right? The disease states. And so the disease states can affect the pharmacokinetics of the drug. Of course, that's why you're giving the drug in the first place. Disease state, trying to fix something, but because the person has got impaired function in some regard, a dose may need to be changed, it may need to be lowered, may need to be increased, if only the drug may need to be used. Right? So diseases that affect the kidney or the liver or the heart, we've already talked about them, right? And just the fact that you get older, right? One thing that's certain is, the older you get, the more likely you are to get something. If something's gonna be on that depressing document one day called the death certificate, something's gonna get you, right? It might be diabetes, it might be cardiovascular disease. It might be old age. That would be cool. But, and my wish for everyone. But something's going to get you, and in the U.S., usually what it is diabetes, or heart disease, or stroke, or the C word, which I don't like to say. Right, so diseases, and so this person's got some challenges going on, so you're bringing the drug into their body, and their body is eliminating it, but is something affecting the elimination? Because if there is, then drug may give an unexpected effect. Okay, and so age, just a simple matter of age, can increase person's likelihood of various diseases that affect important organs. So that's PK related, and then we have I'll go to the bottom here, drug interactions that may cause an unpredictable response, right? An unexpected response. So in a single drug situation, you're taking a single drug, there's no need to worry about a drug-drug interaction. There's just monotherapy, one drug. But in a situation where you've got polytherapy, where you've got more than one drug, now there's a potential for an interaction. Doesn't mean there will be. And not all interactions are equal. I mean, some truly are life-threatening. Some are, tolerable to a certain degree. Right? And so drug-drug interactions can obviously cause variations. And, if you just kind of think for a moment, the average senior citizen in America takes 5 drugs a day. Good for work, good for business, hard for managing the drug in the patient. 5 drugs a day. Some of them will be taken once a day, some will be taken twice a day. Some of them might only be taken once a week, and you're old, and maybe you're forgetful, So that's a possibility. And then thirdly, pharmacodynamic differences. Pharmacodynamic differences can be the result of genetic differences that impact enzyme activity, or in some cases, drugs that impact enzyme activity. So phenobarbital will induce hepatic enzymes, right, which can clear drugs faster than they otherwise would. That would be an effect on enzyme activity. Cimetidine or omeprazole can block P450s in the liver, right? And so instead of hepatic reaction and cleavage and excretion of the drug, eventually drugs just accumulate, and you get an exaggerated effect. So there are ways to get variations due to certain drugs, right, affecting hepatic reaction. And then there are genetic differences. So the genetic differences form the bridge of our discussion into Chapter 5, which is Pharmacogenomics. And so genetic differences are an interesting kind of thing because you can't see a patient's genetic differences. Right? You don't know if the patient in front of you is a poor metabolizer or wild type. You can't see it. What you can see is their phenotype, right? The phenotype is what is expressed. The genotype is the code inside your cell. And there's no way to see the genotype unless you donate blood and somebody analyzes your DNA and sequences it. Right? So you look at Angelina Jolie from a distance, and she's a beautiful human being, but, and very successful and an actress and all of this. You can't see that she's a BRCA mutant. You can't see she's got a mutation in BRCA1 that increases her risk of breast cancer. You can't see that. Right, so how would she know? So she goes in, she gets her blood tested, she's got a family history, boom. She's carrying a variation of the BRCA gene that is associated with cancer, a cancer-associated mutation. Are all mutations cancer-associated? No. Some mutations are just benign mutations. Right? So you can't see your patient's genotype. You can only see their phenotype. And there is some kind of gap between the two. Because even if you have a cancer-associated mutation, it doesn't mean you have 100% chance. Right? Usually the highest incidence of cancer, if you think of cancer-associated mutations, is somewhere in the 80 to 90%. So there's like 10% of people who, even though they have this variant, may not get cancer. So what's the gap there? How is it that the genotype and the phenotype are not in sync all the time? And the answer is it's a mystery. It's not clear. It's the subject of research. Right? But phenotype is what you see. So when we start to talk about variations in drug response, what we see are, wow, plasma levels of this drug in that patient are very out of whack with typical plasma levels after administration of this dose in many hours. Something's out of whack here. I see this. But you don't know the genotype until you genotype the individual. And so, is that like a normal part of your everyday pharmacy thing? Patient comes in and gives you a blood sample and you know their genotype on the spot? No. Right? So some of what I'm going to talk about today is like the pool of theory. We have this knowledge, and a lot of it's like kind of amorphous theoretical, like, okay, polymorphism, not polymorphism. But it hasn't always translated yet into an application that you see in the pharmacy, but I'm going to convince you of how that will happen more and more in the next hour or so. So genetic differences can be the cause of variations in drug response. And one of the key examples of this is a phase 2 reaction called acetylation. Remember, we talked about acetylation as a phase 2 metabolic reaction last class. And we said that acetylation is one of the primary mechanisms of the metabolism of a drug called isoniazid. And isoniazid is a historically important drug. Its name is down here for you. And you may have noticed I switched the order of the slides before class started, so you have the slides somewhere. But I'm trying to build into the pharmacogenetics, and earlier today I was all over the place, but maybe I still am. But anyway, here we are. Right? So isoniazid is like if I could have discovered any drug in my life, it would have been this drug. Because this drug changed humanity, because people with TB survived. Right? TB is like in the U.S., like what? What's that? Right? In other parts of the world, TB is not, what's that? It's like, whoa, 2 billion people infected. Right? So TB is a big burden on the global health, but luckily, because of drugs like this and public health prevention and all the stuff you learned about in class and the second year, it's kind of like a distant memory for us. But you go back 100 years in the U.S., you go back to 1915, and about 1/4 of the population was infected with TB. And out of those, maybe about 1/3 of those patients would die. So they had these places called sanatoriums, which were basically like big halls that were as sterile as they could be. And you had nurses and you had sisters coming around and making sure that the patient's needs were met, that they were fed. But there were no drugs. Or the drugs that were out there were severely toxic. Right? There was this drug called Salvarsan, but it was like arsenic. It was really nasty stuff. So along comes streptomycin. Streptomycin, any of you ever heard of that one? That one was discovered at my other alma mater, Rutgers. Right? So they discovered it in the soil. Some guy was studying soil microbes. And he found that microbe A releases this thing which kills microbe B and gives it an advantage. Well, chemical killed and it's like this microbe in the soil. So then they found out that it killed lots of bacteria. And so streptomycin was an important finding. But the problem with streptomycin was that TB became resistant to it quick. And so you had a lot of streptomycin-resistant TB that wouldn't respond anymore. So along comes isoniazid. So isoniazid, boom, comes out with a revolutionary drug. And today there are isoniazid-resistant strains out there, but it's still used often. And so what is this drug? It's a drug that prevents growth of the mycobacterium. And so another thing about that disease, which I'm sure you've probably learned about in public health, is that you can be a carrier and show no symptoms, not be sick, and actually not be infected either, but it's in you, right? And then there can come a time where the disease pops out and becomes active. It goes from a latent state to an active disease. The active disease is called consumption. It's like a trauma on your body. It's a travesty. It's like ravages you. And so if you ever look at old images of consumption on Google, like be prepared to be depressed because it's like nothing, like a person's skin and bones. The disease has consumed their body, right? So that's an issue. So there is this latent disease, which is the most common type, and then active disease. About 10% of cases become active, of all cases. But the point is, there are people in the U.S. who get isoniazid too. I had a colleague one summer when I was up in Woods Hole. My PhD advisor used to take me with him to Woods Hole. Anyone know where that is?
It's in the state of Massachusetts, and it's in a part of Massachusetts called the Cape Cod. Have any of you ever been there? Some of you. Make sure you go sometime in your life. And go in July. There's like a 2-week period in July where you go swimming in the water, and little sparkles of green light up all around you. It's like being like in another planet. Those are called dinoflagellates. They come in for 2 weeks a year. And you swim and it activates calcium and stuff. Whoa. And so, of course, I was up there with my professor collecting sea urchins into a bucket. That was my job. And then I used to have to inject them with salt and cause them to dump their candy. Inject them with sea urchins. They dump their candy because they think they're dying and they want to reproduce. And so we collected sperm and we used to study how sperm moves. Because my former advisor was interested in sperm motility. Because one of the problems with infertility in men is lack of sperm motility. So sperm swim. They can't make it to their target. And so I used to collect these sea urchins, inject them, dump their gametes. Sometimes I really shouldn't tell you this, but sometimes, but this is an accepted scientific method. You zap with some electricity, and they would dump their gametes. So here I am up there in Woods Hole doing this, and one of my colleagues in the building was also visiting with she was on isoniazid. I said to her, "Why are you taking this TB drug? You have TB." I didn't know if that's like a crossing the line kind of question because I'm very good at that. That's my talent. That's my talent. And unfortunately, so I asked, no, but I work with TB back home in my lab, and I seroconverted. So I became, somehow I got exposed to it while I was working with the lab. And so the infection was quiet, really. But she was put on this drug for a year as a way to kill it before it ever can go active. Sometimes that's called prophylactic treatment. Okay, so the point is, let's continue this story in a moment, but this drug is an important drug, and it was kind of a peculiar one in our discussion of metabolism, because it was like phase 2 first. Remember, it was one of the weird ones. It was phase 2 first. Okay, so I shuffled the slides. We've already talked about, cardiac output and its effect on perfusion of tissues and clearance of drugs. So I'm going to go forward on that. I'm going to go forward on the changes in body composition with age, right? Because we've talked about that. Little child, mostly muscle and water. Middle-aged, average American, some adipose. Older American, not physically active, lots of adipose. And the more adipose, usually the more risk of other problems, right? So body composition can change with age, and we've talked about that. Drug interaction, we've talked about that. And so very briefly, the simplest example of a drug interaction would be a patient taking 2 drugs, where one would induce a cytochrome P450 such as 1A2, while the other would be a substrate for cytochrome P450 1A2. And so in this situation, one of the drugs is inducing metabolism of the second. So the second drug isn't going to work as well as you would have predicted. Decreased clearance of drug 2, because metabolism has been induced. Does that simple interaction make sense? Now, another simple interaction. The first drug may be an inhibitor of 1A2, while the second drug is a substrate. So inhibiting the enzyme that breaks down the drug. Decreased clearance of the drug and perhaps an exaggerated effect. Now, I realize that that's simple, right? And in a patient taking 5 drugs, it can be complicated. Right? But what I want you to understand here at this point is the concept of how you can get at a drug-drug interaction through a PK phenomenon. All right, so there are lots of consequences of drug interactions. One drug might interact with another to impede the absorption of that second drug. you can envision lots of possibilities. One drug may compete with another for its binding to plasma protein sites, which is an important, and that second one is called a displacement reaction, where you displace the drug that was sitting on a plasma protein with the second drug to come on board. One drug might change the excretion of the second, either through kidney or liver. One drug might affect the metabolism of the other. So these are all possible ways of arriving at a simple type of drug-drug interaction. Now, it's true, like, if you practice and, you're dispensing some drug, and the patient's already on another drug, and a red flag comes up on your computer. Have you ever seen that? Right? So some of those drug-drug interactions are potentially very serious, and you need to follow up by calling the doctor. Some of those are going to be things that the doctor may have told you are under her control. She's watching it, right? And so that kind of knowledge will come to you more in the therapeutics curricular courses. When you get to drugs and disease, the way that course works is you have the pathologist, and then you have the med chemist, and then you have the pharmacologist. We talk about all the problems. And then the clinician comes in and then kind of sets the story. Well, here's patient X, and he's got high cholesterol, and he's got heart failure. He's got diabetes, and he's got all these problems. And, the ideal drug for his heart failure is not useful because it's got renal disease. And so we got to try something else. And so it gets even more fun, I think. Not for him, but, for you, and understanding how to go about managing him. But here our job is to just get the concepts there so that when they start talking about this stuff, you can kind of piece it together. All right, so mixed function oxidase system we've talked about last class. We've talked about the location of these reactions. So again, I'm going to try to cut to the genetics sooner rather than later. All right, we've talked about this. Again, I've shuffled the slides, but nothing new on this slide. The CYP superfamily. The only thing I can maybe add to this that I haven't yet said is that while most of the mixed function oxidase class is made up of the CYP superfamily, and most of the CYP enzymes are in the liver, there is one other site where you can find a lot of P450s, and that is in the gut. Okay, so the primary extrahepatic site, primary site outside of the liver where you can find P450s is in the gut, heterocytes. And so again, we alluded to this last time where a drug might be metabolized as it's being absorbed through the intestine. So that can also occur sometimes. So if you're kind of ranking them, the liver is like way up there in terms of market share of CYPs. And then the next level would be the gut. Now, in years past, I've kind of talked about how CYPs are named. We have these weird names like 3A4 or 1A2 or 2Z6. Where do these names come from? So the answer to that is that the names and the categorization are based on sequence homology of the various members of this general class. And so in order to be in the same family, like to be a CYP1 or a CYP2 or a CYP3, to have that as your first number, you need to have more than 40% of sequence homology with the other members. Now, I'm not going to really go deeper into this because I've learned my lesson. It's not always so easy to follow. But if you want to exactly know how the naming schemes have been arrived at, it's due to sequence homology. So you put all the similar looking ones in a box. You put all the other similar looking ones in a box. And then, I've got 3 that look similar, so I'll call them 1, 2, and 3. Or the box will be a different letter. Right? So I got box, 1A, I got box 2D, I got box 3A. That's helping you. If it's not helping you, then just realize that there's a kind of a peculiar naming system where you have an Arabic number, a letter, and another Arabic number. Okay, and there's a few of these names that we need to know, even though they're kind of weird. And so the major isoforms of cytochrome P450 in humans that drive 90% or 99% of drug metabolism are CYP3A4, 2D6, 2C9, 2C19, 1A2, 2A6, the toxic one, and then this one's listed twice by accident. Right? So these are the primary CYPs in humans that contribute to metabolism of drugs. Now, dietary considerations. There are certain components of a diet that can affect CYPs. The first of these I'll mention is grapefruit juice. Grapefruit juice contains a component that is an inhibitor of CYP3A4. So grapefruit juice is like a drink derived from a plant, right? The plant has given us a fruit, and the fruit is smushed up and diluted with a little bit of water, a little bit of sugar, and there's your juice. But the juice contains lots of compounds. It's not just like nothing in there. There's lots of compounds in the juice. One of its components of it is a 3A4 inhibitor. And so there were people taking drugs metabolized by 3A4 that were starting to show like big accumulation in the body, lots of exaggerated toxicity. The physicians tried to figure out what's going on. And they found out that in some of the patients where these things were happening, they were drinking grapefruit juice while they were taking their drugs instead of orange juice or instead of water anymore. So I don't know if you've ever seen this on your prescription, but sometimes it will say sticker like, I'm not taking grapefruit juice. This is the reason. It means it's metabolized by 3A4. Although grapefruit juice can affect that. Now the truth is, it's usually not a cup a day of grapefruit juice that will do this. It's usually like 6 or 7 cups a day of grapefruit juice that will do this. But generally, the idea is to avoid it. And you're probably too young to remember, but there used to be this grapefruit diet that was kind of marketed a lot. And it actually kind of works, because I don't know if you've ever had a grapefruit. Like, do you feel like eating in the short time thereafter? Like, I don't. It works on me. And if I don't want to eat, I choose grapefruit. So that's one kind of dietary effect, dietary drug interaction. Another has to do with vegetables. So certain types of vegetables that can be categorized as cruciferous vegetables, these are vegetables of the cabbage and broccoli and Brussels sprout variety, cauliflower. I love cauliflower. Oh, it's like the best. And it looks like brain. So it's like even academic. But anyway, all of these contain components that upregulate a type of P450 called 1A2. And so too does barbecued meats. I don't know if you like barbecued meats, charcoal broiled meats, out on the grill, it's Memorial Day weekend, you throw in your hamburger, your hot dog, get that black stuff on it. That contains compounds called polycyclic aromatic hydrocarbons, which are able to come into your liver and induce 1A2. Which, by the way, polycyclic aromatic hydrocarbons are also found in the cigarettes that some of you may smoke. Which probably, if you could stop, it would be good. Right? But if you have patients who smoke a lot, their 1A2 levels are higher in their liver than compared to the similar kind of patient who's a non-smoker. Okay, so drug-food interaction. Third example is calcium present in dairy products like yogurt or cheese or milk. You ever see that one? You're not taking dairy products on the label. The reason why that is put on the label is because calcium, in some instances, can act as a chelator of a drug that's inside the lumen of your gut before it's absorbed. What is a chelator? What is chelation? It means claw, right? Claw. Chelator is a molecular claw that binds something, and it prevents it from being absorbed. So it binds to it and blocks its absorption inside the gut. And there's a few drugs which are particularly sensitive to calcium chelation. Tetracycline antibiotics or quinolone antibiotics are two of those. And that's what calcium can chelate these drugs inside the lumen. And that would lead to decreased absorption, and that would lead to decreased efficacy because the drug's not getting in to have an action. No, I believe the compound in grapefruit juice is there in the actual food. So like that's it. And it's not in grapefruit. It's grapefruit. Citrus, but only grapefruit, not orange. But that's a good question. Actually in the fruit as well. Okay, age. We're going to go through that quickly because we've talked. Genetic factors. Okay, here we go. We're starting our genetic discussion. So genetic polymorphisms have been found in these various drug metabolizing enzymes. In 2D6, 2C19, and 2C9, and 2A6. Those are all, by the way, phase 1. And N-acetyltransferase, which is a phase 2 enzyme. So let's put some definitions out there. The word polymorphism. Poly means many, morph means forms. Many forms of a gene. So in a typical cell in your body, we have two copies of the DNA. One we got from mom, one we got from dad. And those DNA molecules contain genes on them. So in a particular drug metabolizing gene, if we're talking about a drug metabolizing gene like 2C9, you get one copy from mom, you get one copy from dad. Now, if there's more than one possible allele in the human genome, you might get a copy from dad that's different from mom. Or you might get exact copies. So let's step back for a moment. Think back to where you are in your family line. Here you are, it's 2015. And your parents had you, their parents had them, and their parents had them, and their parents had them, and their parents had them. So you could probably trace your history back to 300 or 400 years. Really think about it, right? Or more. So the thing is, all this time, you have life in action. Right? You have cells growing and dividing. Right? And so when dad's cell meets mom's cell, which by the way are gametes, so they all have one copy only, they merge, and they become you. You have two copies. And so that moment is wonderful because here you are, you're on your way. Right? Journey begins. But you're so tiny that you could never see yourself, even if you had eyes to look at yourself at that moment. Right? So you go from tiny itty bitty you, I don't know, how old are you, 20, 25? Let's say you're 25. You go from tiny itty bitty you to now you at 25. You've gone from two cells merging into one to maybe 1 trillion cells being formed now. You've gone through mitosis. What's happened in replicating your DNA? What enzyme replicates DNA? DNA polymerase. So DNA polymerase, so when the DNA unwinds during the S phase, right, of the cell cycle, the polymerase sits down on one side and uses it as a template. Remember this? It's called the replication fork. You learned about that, right? So the polymerase uses the one strand as a template, and it makes a copy. So if there's an A on the template, it puts a T there. If there's a G on the template, it puts a C there. If there's a C on the template, it puts a G there. And it does it for 3 times 10 to the 9th bases in every strand. Do you think that it's absolutely perfect and never makes a mistake ever? It makes a mistake sometimes. Does it make a mistake a lot? Like all the time? No. We call it a high-fidelity enzyme because it's very faithful to the strand. Right? High fidelity. Like, you would probably prefer to have a spouse who's completely faithful rather than one who's highly faithful. Right? Like, high fidelity is awesome, but complete fidelity is like, now I'm ready. I'm not scared anymore. Right? So I digress. Into the philosophical. But so high fidelity means it's pretty good. It's going to make a good copy most of the time, but every now and then it's going to make an error. So let's imagine that this DNA polymerase has replicated 3 times 10 to the 9th base pairs, and it made one mistake. What's the percent error on that? 1 over 3 times 10 to the 9th. It's not very big. So tiny. I'm not even going to say what happens if you completely faithful. The partner makes you tough. But a DNA polymerase, high fidelity, makes one mistake. Okay, one mistake out of 3 times 10 to the 9th. That's pretty good. Right? But the thing is, if that one mistake is in an important site of a gene, it can affect that gene's function in the next generation. Right? Because that one mistake now becomes the copy template for the next mitosis. Yes? For the next replication. So the DNA polymerase just replicates. It doesn't know if it's replicating the original sequence or the altered sequence. Its job is to replicate. So once there's a change put in during evolution, it stays. Unless there's some reason to get rid of it. Unless it's non-advantageous. So if you think back in your germline long ago, when you went from, when your great-great-great-great-great-grandmother went from 1 cell to 2, and from 2 to 4, and from 4 to 8, and from 8 to 10 billion trillion cells, there may have been a mistake somewhere. An inefficient copy that got carried into her germ cell and got transferred to you. And so the thing is, when you go all the way down the strand, and you almost make it perfect, but there's a single change. It's called a single nucleotide change. Single nucleotide polymorphism. Another form. So yes, we all have genes that we need to survive. And many of our genes look exactly the same. Exactly. Nucleotide to nucleotide. But every now and then you come across a polymorphism in the gene. So your mom's gene for CYP2D6 might look almost completely like your dad's gene for CYP2D6 that you inherited. But it's different enough that it did for metabolized copies to affect the coding of the eventual coding. Remember, the gene codes for the message, codes for the protein is the enzyme. So this idea of polymorphism means many forms. So you can have cytochrome P450-2D6, and in the ideal world, there would only be one copy in every single person that would be identical. But because of all of this DNA replication and mitosis throughout the thousands of years, there's been some mistakes, some changes in the Sometimes the changes are highly advantageous. They give you a survival benefit. Sometimes the changes are benign. Sometimes the changes are deleterious. And if they happen to be seriously bad, usually the progeny doesn't survive the pregnancy. Right? So if you have some serious genetic error that happens in that developing baby, the baby might never make it after pregnancy. Right? That's why I always get nervous when I see my friends putting on like the ultrasound pictures in the first month. Come on, people, a lot of those pregnancies end before they make it into the third month. So, I call it, temper your joy. Just just wait. Usually if you clear the third month, your odds of making it to the end are very good. But, everybody's going to do what they got to do. But I always cringe when I do that. I don't know. I kind of think it's a personal thing. I think you should know what you're saying. But, people are into it. Who am I? Not good. But the thing is, mitosis from this little thing to you. Potential for changes, potential for errors. So polymorphisms mean changes, multiple forms. So you might have a gene for 2D6 that now if you're to look into all of humanity, right? You look into Asia, you look into Africa, you look into Australia, and you look around the globe, you find that, hey man, there's actually 12 forms of this thing in humanity. And most people have the original copy, but some of us have slight change. Called single nucleotide polymorphism, or sometimes kindly referred to as SNPs. Single nucleotide polymorphism. So this idea of the polymorphism means more than one form. Now, in truth, polymorphism means more than just a change. A polymorphism is actually a change in a gene that's found in greater than or equal to 1% of the population. And greater than or equal to 1% of the population. 1% of the population doesn't seem like it's that common. Right? 1%, 99%. But if you think about the population of the U.S. right now, 330 some odd million people, 1% is 3.3 million people. Right? Now expand that out to 1 billion people in China. I can't do that. 1% of a billion. Is that a million? I'm embarrassed. I can't think of that. Right? A lot of people, right? So the percentages, when you kind of scale up to the population, could represent a lot of people. Now, usually, if there's a change that's in less than 1% of the population, right? So a SNP in greater than 1% of the population is a polymorphism. A SNP that's found in less than 1% of the population is called a genetic variant. So a genetic variant means that it's different from wild type, but it's not very prevalent in the human genome. So to give you an example, when I was at Rockefeller, my postdoc, I worked on a genetic variant of BRCA1. Genetic variant means less than 1% of the population. So my mentor was friends with a mentor from Sweden who's an oncologist there. And there's a connection between Sweden and hereditary breast cancer. They have a lot of what are called founding mutations, or these polymorphisms that increase the immune base of the breast. So he called up my mentor and he talked to my mentor about this genetic variant he had seen in one of the patients and couldn't find it anywhere. So what oncologists do is when they biopsy those tumors, they have them sequenced for various genes. And those changes that they observe in their patients, they can put into databases. And then later, the physician will update the database as to the outcome. So, the patient with this mutation survived greater than 10 years. The patient with this mutation didn't survive greater than 6 months. The patient with this mutation survived 25 years. Whatever it happens to be, they update this information. And they form these databases that are based on genetic variants and genetic polymorphisms. So, if you have a genetic variant, it's going to be hard to find it in the database because it's very rare. You're not going to have any idea what the true risk of change is. But if you have a genetic polymorphism, that's not going to be so rare. So when Angelina Jolie decided to have a blood test to see what her BRCA status was, they put it into the database and they pulled out a mutation that is associated with cancer in most patients have it. She didn't have cancer yet, so she had a mastectomy based on genetic tests to prevent development of the cancer. But that mutation correlates with a high likelihood. That's called a cancer-associated mutation or cancer-associated polymorphism. There are many polymorphisms in databases around the world that are what we call benign polymorphisms. They're different, but they're not associated with individual staff. Right, so instead of having a green Maserati, I got a yellow Maserati. Maserati, drive around that. What's it? Looks really good. Benign change. And so I got assigned to study this genetic variant, which has, to the present time, only been observed in one family in the whole world. And the irony of life, the patient who had the change died from breast cancer when she was 18. Very aggressive cancer. And her twin sister was completely healthy and had 2 wild type. Right? So we did studies. We found out that the change was an important region in the protein. You know, we published that and we moved on. But the thing is, when you study rare variants, in the current economic climate of funding, the funding agencies will come to you and say, well, you know, Dr. Galak, you're really studying like one person's problem. And we want to invest this money in a project that studies all people's problems. But you're studying a rare thing. Why don't you study a polymorphism? At least it's 1%. Why are you doing this rare thing? And the answer is sometimes you just do it because you got to do it. You have a purpose and you got to do it. But this brings us to this idea of the one versus the many. Right? The one versus the many. So what I'm talking about today in these polymorphisms and their implication on DNA metabolizing enzymes, we could kind of talk about this with breast cancer susceptibility genes like BRCA1 or P53. You know, we could talk about this for other kinds of genes. But here we're applying the discussion to the genetics of genes involved in drug action or in drug metabolism. So that's why this field is called genetics, but it's given a qualifying prefix called pharmacogenetics. It's the genetics of systems that affect drug action or drug kinetics. It's not all genetics, it's the whole organism. Yes? So pharmacogenetics, under the big umbrella of genetics, is looking at the genetics that affect drug response. Okay, I was going somewhere with that, but I don't know where. Where are we going? Where are we going? No. All right, so if we look now at these genetic polymorphisms, and we start to look at certain groups of patients, certain trends start to show up. So, for example, if you were to look at Caucasian population, 5 to 10% of Caucasians estimated to be poor metabolizers of drugs for CYP2D6. In the Asian population, poor metabolizers of substrates for 2D6, 1 to 2%. However, in the Asian population, up to 20% of the people are poor metabolizers for CYP2C19 substrates. Now, first of all, let me qualify all of this. We're not picking on any ethnic group in particular. This isn't like we're just comparing ethnic groups for the sake of comparing ethnic groups. Right? But what I just told you, right, about these polymorphisms back to front, right? We live in an age where we travel. It's easy to move, right? You can get on a plane from JFK and be on the Rome campus of St. John's in 10 hours. You don't have to take a boat across the ocean and get sick from the waves and have people throw up on you, right? You don't have to do it. You get on a plane and you're in Italy, and then the next day you can fly to Germany to go to some skiing event, and then you can fly to Moscow to see Vladimir, right? And then you can just keep going, right? So the world is different. But if you go back in time, like 100 or 200 years ago, yeah, you had immigration. It's true. But it wasn't like now. So if you grew up in Southeast Asia, you probably married a girl from Southeast Asia. You probably had kids with a girl from Southeast Asia. And that kid probably married a girl from Southeast Asia. And so you had, familiarity, right? If you grew up in Africa, you probably married a girl from Africa. You had a kid in Africa who married the daughter of someone else who grew up and had a kid in Africa. Right? If you're from Australia, you probably grew up in Australia and had a kid with somebody from Australia who had a kid, who married and had a kid with another person from Australia. So the reason why I'm talking about this is because there's something called genetic drift. Right? There's like a noise associated with the genes. So you might see a single nucleotide polymorphism in an ethnic group because somewhere back in all of these cell divisions and DNA polymerase activities, somewhere there was a change that stuck. It just kept getting propagated. So you can identify SNPs in certain ethnic groups. That's how they do that. Like when they give a blood test and you try to find out, like, are you Egyptian from the Pharaoh time, or were you on the Mayflower? Right? They give you a DNA test and they tell you 8% European, 90% African. They have these tests that you can do. How do they do that? They base it on SNPs. But you see, the world is changing now, so these numbers will not stay this way. Right? Nowadays, you have someone from Australia coming to the U.S. and marrying someone from the U.S. You have somebody from China going to Asia, going to Africa, and getting married and having kids there. And that child's marrying somebody who's the product of a Canadian/French union, right? The world has changed, so we're getting genetic mixing, and that's a good thing. There's nothing wrong with that. But the thing is, people in the general public, when they see numbers like this, they feel that the numbers are being used to put somebody down or to say something negative about a certain ethnic. So you're a healthcare provider. Knowledge for you is neutral. But realize that all knowledge has potential for good or for bad, right? The people who developed the atom bomb, right, in the Manhattan Project, they were interested in the physics of nuclear radiation. They didn't want the bomb to be dropped in Japan and kill all these people. That wasn't their desire, but that's what happened. But that wasn't their desire, right? So knowledge can be used for good, could be used for evil, but knowledge itself is neutral. Like, the desire to know is just a human thing. Like, you want to know. I want to know, right? And so the knowledge on ethnic groups and genetic prevalence and whether somebody's a poor metabolizer or a fast metabolizer is not necessarily a bad thing in any way, but it can be misinterpreted by people. So as a healthcare person or scientist, you need to educate people when you hear things that are not true. Okay? Yes, it's true that if an insurance company finds out you're a poor metabolizer of clopidogrel, right, and you need to use some other more expensive drug because you can't metabolize it to the active form. That's actually not a drug, it's a prodrug. So remember, a prodrug is metabolized to an active form. So if you're a poor metabolizer, like many Asian people are, of clopidogrel, they can't activate that drug. That drug can't have antiplatelet action. They have the stent put in, and the stent gets blocked. And the person ends up in the hospital. That's no good. Right? No good. Because what's going on? You have another ambulance coming. You have potential for death. You have another surgery. You have another nurse. You have another surgical tech. You have another doctor. You have another test. And that's going to cost thousands and thousands of dollars. So, I'm raising these issues because in some cases it would be good to know if you were a poor metabolizer before they gave you the drug. But you look at the patient, I can't tell just from looking at them if they're going to respond to the drug. So, we're swimming in the pool of theory today. But here's an example where this could very practically have an impact if all hospitals decided to genotype their patients before giving them plasma. Right? That kind of approach is being done today with certain other drugs. So there's a drug called 6-thiopurine, an anti-cancer drug. It's used to treat certain kinds of leukemias. And it's a great anti-leukemia drug. The problem is it kills the tumor cells, right? And in some patients who are poor metabolizers of it, the drug accumulates to levels where it now starts to kill bone marrow cells. So when you kill the bone marrow cells, you start to kill the red blood cell precursors. So what happens to the patient? They develop the AIDS, anemia. You start to kill the white blood cell precursors, and they develop the eye disease, immunosuppression. Right? So all of a sudden, that drug that worked great to control the tumor now it caused them to be immunosuppressed. Now caused them to become anemic. Now they acquired some MRSA infection while in the hospital, and now they may die because they're a poor metabolizer of the drug that helped them start to be. You see the irony in that? So places like Fox Chase Cancer Center in Philly, they're going to genotype their patient before they give them that drug. And if they see that patient's a poor metabolizer, they're going to not give them that drug. They're going to try to give them different so that their patient avoids immunosuppression, so that their patient avoids anemia. So while, yes, today, 2015, we are kind of swimming in the theory pool, some of this is starting to hit practice. And you all, most of you, are going to be working for the next 45 years before you retire. Right? So it's 2060. I think you're going to see these things more and more.
Hopefully I'm still around. I'll be pretty old, but you know, God willing, you can tell me about the pharmacogenomics things you're doing over a cup of coffee. Okay, so are we following this discussion? Right. This slide, enzyme induction in the liver, I'm not going to talk about. Running out of time. I'm not going to talk about this slide, but I've kind of said this stuff. Okay, pause. Classical inhibitors of P450 include cimetidine, which inhibits 3A4 and 2B6, ketoconazole, which is the global P450 blocker. Now, ketoconazole is a systemic antifungal, and usually the patient's on it now, and in the hospital, you know, other drugs are failing. But cimetidine is over the counter. You can buy that over the counter. So maybe your patient's taking drug A that you're dispensing for her, but she's self-medicating on over-the-counter cimetidine, and something weird happens. She's got a tremor. She's got a rash. You know, so it might be worth asking, is there anything you're taking over the counter, just so you kind of have an assessment of whether there's some kind of other thing happening. Like non-prescription agent. It used to only be by prescription, but once the drug goes from prescription to over the counter, pharmaceutical company is happy or sad. From prescription to over the counter. So happy. You don't need a prescription. You don't need to go to the doctor. You just go and buy it. And now I'm going to put commercials everywhere, and I'm going to get you to think you want it. You're going to buy a lot of it. Once you go from prescription to over the counter, man, you go from like $10 million drug to $50 million drug. So always fight to keep it behind the counter because that's your job. That will keep you working forever. And also is safer. I might delete that part when I put this up. Okay. Okay, good. Okay. 3A4. 3A4 is a big one, man. This one is like the Alex Rodriguez of cytochrome P450s. Right? It's like, look at all of these substrates, man. And guess what? They don't even all look the same. Like, I don't know, here's a steroid, prednisone. Here's an azole, ketoconazole. There's an amlodipine, that's not that. Cocaine and opioid. They're all kind of different, right, in their structures. You might not remember their structures. But trust me, there are some very different looking structures in this list of substrates. So do you remember what we said about P450 active sites? They're peculiar because they're very permissive. They let lots of substrates in. That's not true of most enzymes. Most enzymes have a particular substrate in mind and that's it. Here we've got a lot of, sometimes they call it a promiscuous active site. A lot of possible interactions. Right? So here is cytochrome P450-3A4. And here are some known inducers of P450-3A4. And there are multiple ones. But here's our classical phenobarb, our phenytoin, our rifampin. Those are, like everybody knows those induce P450. So you should know it too. And then on your homework sheets, and I know this is now after the midterm test stuff, I ask you to know a few more. Here's our grapefruit juice constituent that causes P450 inhibition, as well as some of these other agents like ketoconazole, like cimetidine. So the ones I've kind of pointed to are the ones that you definitely should keep in mind, right? Inducers like phenobarb, like rifampin, right? Like phenytoin. Inhibitors like cimetidine. Inhibitors like grapefruit juice for some of them. Yes? Okay, very good. Awesome question. I'm glad I didn't go too far ahead. So, yes, some drugs induce their own metabolism. And those kinds of drugs need to be monitored. And this is an example of that kind of drug. So, carbamazepine is an anticonvulsant drug. I don't know how prevalent its use is. I know it's Dr. Scalante's favorite drug. He studies it in the lab a lot in his research. And so here's an interesting drug. If you give a single extended release dose of the drug, the average half-life is about 36 hours. But upon continuous use of the drug, look how the half-life changes. Right? This drug has a much longer half-life before it induces its own metabolism. When that happens, the half-life falls. It's more rapidly cleared. There's more P450 that's taking it out. Yes? So to answer your question, at this point, the dose has to be adjusted for the drug medication. Because blood levels will fall unless you increase the dose. So, yes, some drugs induce their own metabolism, and you need to keep an eye on them as the patient goes through treatment. All right, here's our cytochrome P450-2D6. And again, we have this mega list of stuff. Right? And I'm just going to call your attention to this drug, imipramine. Imipramine is a tricyclic antidepressant. And in 5 to 10% of Caucasians, there'll be a poor metabolizer of that drug. Okay, is nortriptyline on the list? There's our nortriptyline. I'll talk about that. So keep those in mind, those two drugs for the moment. And keep in mind that this is 2D6. And now look here at 1A2. What do you see? Imipramine. This is meaning to show you that one drug can be metabolized by more than one P450. One drug can be metabolized by more than one P450. So when you open the package insert, it might say, this drug is primarily metabolized by 2D6, with about 10% of the dose being metabolized by 1A2. It might say that. So one drug can be a substrate for more than one P450. So not only are the active sites promiscuous, drugs are promiscuous from one enzyme to another within the family. Does that make sense? All right, if we look at 2C19, I would just add to this list Plavix, which is also known by its generic name of clopidogrel. It's not on the list. Gives you an idea of how old this list now is. But anyway, the 2C family, highly polymorphic in Asian populations. So let's go back to our isoniazid example. Isoniazid gets metabolized through an acetylation reaction of the nitrogen here, the amino group. That's why it's called N-acetyltransferase, N-A-T, or NAT. NAT2 is actually the name of the gene that codes for this enzyme. So follow the amino group, NAT acts, boom, acetylated. The enzyme that does it is N-acetyltransferase. Now, if you look at plasma levels of the drug versus number of people that have those particular plasma levels, you see actually, in the case of isoniazid, a bimodal curve. Now, you don't see a normal distribution, right, where everybody's kind of around a single average. But rather, you see a peak here, You see a peak around here. Right? That's indicated by this light gray and dark gray. The light gray have much shorter half-life of the drug because they're inactivating it more quickly. Then these patients, the dark gray, have a much longer half-life of the drug because they are slow inactivators. Okay, so this kind of observation is an example of monitoring the phenotype. Right? How the person's body responded. You don't know based on this the genotype. These were the first studies. They came first. People predicted genetic changes based on phenotypic changes. Right? This study was actually done in like the 1960s. It might be the next slide. It's not, actually. So I'll get back to this in a moment. The next slide is the definition. So the definition, pharmacogenetics versus pharmacogenomics. In pharmacogenetics, genetics pertaining to drug effects through the eyes of a single gene. So actually today I've had a lot of small discussions about different metabolic enzymes, right? Different genes involved in drug metabolism. So each of those discussions is considered a pharmacogenetic discussion. I've talked at one gene at a time. I've talked about 2D6. I've talked about 3A4. I've talked about 2C9. One gene at a time. Pharmacogenomics is now looking at all of the possible alleles in the whole human race and all of the metabolic enzymes. So the basket is much larger. Here we're looking at multiple genes, multiple polymorphisms, which, by the way, is the patient, right? The patient is multiple things. Right? So we have this concept of pharmacogenetics, which is one gene at a time affecting drug response. Pharmacogenomics, which is multiple genes together affecting drug response. So in my approach to this subject, as a simple approach, it's much more easy to understand one gene at a time and effect than to understand a panel working together, metabolizing rapid and ultra slow all together. It's a harder thing to understand. Right? I think in the world of pharmacogenomics, people don't even understand it. They think they know what it is. Right? Or maybe that's condescending. Maybe your definition of it changes. Maybe you familiarize yourself more with it. Maybe that's what it is. Maybe your definition gets more mature. Maybe that's what it is. But one definition that kind of works for me is the molecular study of genetic factors that determine drug efficacy and toxicity. So all of the multiple genetic factors that can impact the success of that drug in the patient and try to hopefully minimize the risk. All right, so last night I posted a link to an article called Pharmacogenetics and Metabolism Past, Present, and Future. And it's a free open access article. And it's 27 pages, but you can probably just print the figures. It's about 5 figures. And it kind of puts it all together here. Here we have the situation where we have a responder to the drug. They take an active drug. The phase 1 enzyme acts on the drug to form an inactive metabolite. Right? That's our typical model. Then we have a situation where we may have an enzyme involved in metabolism, phase 1 enzyme, that has reduced activity or no activity. So, okay, by the way, you have 2 copies of every gene. In every cell of your body is a different gene. You have 1 copy of every gene. So here come the 2 genes in your cell. One is from mom, one is from dad. One is normal or rapid metabolizer, the other is normal or rapid metabolizer. What's your phenotype? Normal or rapid. Those are synonyms. Okay, here's gene from mom, poor metabolizer. Here's gene from dad, poor metabolizer. What's your phenotype? Poor metabolizer. Here's gene from mom, functional, rapid. Here's gene from dad, poor and slow. What's your phenotype?
Well, it's usually something called intermediate grade metabolism. Okay? So when you see these terms in Chapter 5, poor metabolizing, rapid metabolizer, intermediate metabolizer, it's talking about whether you are 2 good copies, rapid, 2 defective copies, slow, or 1 good and 1 defective. And then there are some exceptions. They have ultra rapid, sometimes ultra slow. So try calling someone ultra slow. You're ultra slow. Right? That's not, don't do that. So here's a situation where patient is a reduced function phase 1 enzyme. So maybe this is clopidogrel. Right? And so, oh no, wait, that's this one, sorry. So this is an active compound. Right? Active compound, give it to the patient. The enzyme activity is low or no. And so there's not too much metabolite. And there is a lot of drug in the body that's accumulated. And the rate of elimination goes down because it's not being metabolized. And the half-life goes up because it's not being metabolized. And the amount of distribution goes up because it's staying in the tissue and not being metabolized. And so there's tension. In the third case, here we've got a situation, we've got non-responders. So here we're giving them a pro-drug. This is the clopidogrel example. We're giving them a pro-drug. It by itself is not active, so it gets metabolized to an active form. But in the patient that has a poor metabolizer phenotype, the enzyme can't convert it to the active form. So most of it stays in the inactive form, and the person doesn't respond. You see how this applies to clopidogrel? You give the poor metabolizer clopidogrel, it needs to be activated, but it's really not being activated because they've got a poor metabolizing enzyme. And so, the drug's not being activated, the platelets are aggregating, the platelets are forming a clot, hence it's being blocked. This is sometimes called clopidogrel resistance. The patient is resistant to the drug, it's not working. Yes? Do you feel you can tell your patient what clopidogrel resistance is? I don't know if you're going to say you're a non-responder or poor, but you'd come up with a way to tell them something? Okay, good. Better you than me. I don't have to tell them anything. I just have to tell you, and you'll find a way to tell them. Now here's a situation where we have a prodrug coming through normal and being formed into a toxic metabolite. And the toxic metabolite can't be phase 2 conjugated to be inactive. And so the toxic metabolite causes toxicity. So here's an example where the phase 1 enzyme's fine, the phase 2 enzyme's not good. Phase 1 enzyme causes toxic metabolite. Which phase 1 enzyme is good at that? 2E1. 2E1. Right? So there's a situation where that can happen, and isoniazid is falling more into this category, panel D. So again, in the world of isoniazid, this was the actual paper published in 1962, so it's like 50-something years old. And they reproduced it in this article. And so what they looked at was plasma concentration of drug after administration of a dose. Okay, and they looked at a certain time into the study, maybe it was 6 hours, I don't remember. But what they saw is we had this big peak here. So some people didn't have very much in the plasma. Okay? Some people had about twofold more than they did in the plasma. And then some people had as much as 10 to 12-fold more in their plasma than they did. So the author, Dr. Price Evans, is actually the one who came up with this idea of fast metabolizer, slow metabolizer, and intermediate metabolizer. That's where these words come from. Now, to just give you an idea on net N-acetyltransferase, this is one of the tables from the author. And it's listing for you the fact that there are over 14 different alleles of this gene in the human genome. Right, so whenever you see NAT2, that's the name of the gene, N-acetyltransferase 2. That's the one involved in the liver and drug metabolism. Asterisk means allele. So they've named these alleles different things, all the way up to 14b. The first part of the table is not here. It's on your page, but we're starting at 12a. Right?
And so if you look at 12A, what is that allele? That allele codes for a single nucleotide polymorphism. The A at nucleotide position 803 of the gene was changed to a G. Right? So A, if there's a copy coming down this way, what is the copy going to be of an A? C. What is the copy going to be of a G? C. So it propagates, right? Propagated a change, a single nucleotide change. In this particular allele, that single nucleotide change causes a change in this amino acid. It changes from a lysine to a arginine at the 268th amino acid of the peptide, of the enzyme. Why is this number 3 times lower than that number? How many nucleotides code for an amino acid? How many nucleotides code for an amino acid? 3. Right? Triplet codon. Right? So here is our A. It's changed to a G. That resulted in a single amino acid change of a lysine to an arginine. And that's a charged amino acid, and that's a charged amino acid. So what do you feel? You think that that's a big deal, that kind of change? Charge to charge? No, it's not a big deal. Charge to charge should be okay. And look at the phenotype. Rapid. So what kind of polymorphism is it? Is it a deleterious polymorphism, or is it a benign polymorphism? Benign polymorphism. Let's go down here to this patient that carries allele 14N. G at nucleotide 191 changed to an A. Alright? So on the copy, instead of having a C, we're going to have a T. It's a change. It's heritable. Right? So it stays. This results in an amino acid change of an arginine at amino acid 64 to a glutamine. Which of the two is charged? Not glutamate. Not glutamine. Arginine. Right? Glutamic acid, yes, but this is not a D. This is a Q. So, charged amino acid to a neutral amino acid. Is that a big change when you go from charged to neutral? So what does that do to the activity of the enzyme? Slow. It causes it to be less efficient. Does that make sense? Right? So, do you feel comfortable reading a mutation? Could you read a nucleotide mutation from left to right? Which is the normal, which is the one in the wild type copy? The one on the left or the one on the right? On the left. Which is the mutation? The one on the right. At which nucleotide in the DNA sequence? The number in the middle. Right? So if we go from here now to the amino acid, which is the wild type amino acid? The one on the left or the one on the right? The left. Which is the change? The one on the right. Which amino acid residue is different? Number 64. Right? So sometimes, like in allele 12A, you can get a benign polymorphism that doesn't affect the function. In the case of allele 14A, you actually get a significant kind of substitution, right? A missense mutation, an alteration, like a polymorphism that has changed from a charge to a neutral, which is a big deal and which has affected activity. Going through these slides, and again, I'm not going to read them and even expect you to know everything, but if you look, for example, up here at cytochrome P452C9, you have these different alleles out there in the human genome. So let's look at allele 2. An arginine should be at amino acid position 144, but what's there? Cysteine. An arginine is what kind of charge? Cysteine is what kind of charge? Negative. Positive to negative, is that a big deal? Yeah. Think about yourself when you go from being positive to negative. That's a big change. Now, negative to positive is a big change, but it's a better one. It'll keep you out of the antidepressant shelf. Can't believe how much risperidol is prescribed in the bottles of it, like on the shelf. Go thinking of it? Oh, go to the cockpit. Like, whoa, I'd rather just have a hot cake. But anyway, what's the consequence of this? Well, look at warfarin. Variability in warfarin therapy, elevated risk of severe bleeding. No good. So, are you going to monitor your patient on warfarin anyway? Yes. But you're going to see changes in their coagulation state. Oh, something called an IMR. You can measure that. Tell you about how strongly you're inhibiting coagulation. But that's not good, and we can see that in people that have this particular polymorphism. If we look at 2C19, all right, 2C19, this one, inadequate response to clopidogrel. Right? So if you're a poor metabolizer and you have a mutation, like an arginine going to a histidine in this particular allele, but anyway, if you have a mutation that affects the function, all of a sudden now you have a potentially serious undesired outcome. Now, is this common? No. Right? It's not common. These SNPs are rare. But when you're looking at a population, a little bit of rare here, and a little bit of rare there, and a little bit of rare there, means not that uncommon, really, when you expand these frequencies out over 300 million people. So, isoniazid, going back to my friends who have to take this as a prophylactic. What's the big deal with it, and who cares? Well, 1 out of 10,000 develop irreversible hepatic failure, and unless you have access to a transplant, it usually means death. Now, to 1 out of 10,000, that's not that bad. I only got 1 in 10,000 risk. Seems pretty low. But put that now in potentially 2 billion people who have latent infections. Divide 2 billion by 10,000, and you have a lot of patients who can be potentially injured to the point of liver failure. Right? So that's an issue. And so if you have a patient taking isoniazid, which organ function test are you going to keep your eye on? Which organ do you want to know is working well and not being injured? The liver. Right, so have you ever heard of something called LFT, liver function test? Right, looking at certain serum enzymes. So usually they shouldn't be there. But if the liver's injured, now all of a sudden they're there. Okay. So let's go back to the drug just as an interesting example. Isoniazid is acetylated by NAT2 to an acetylated form, which gets hydrolyzed spontaneously to this compound, acetohydrazine, which usually gets acetylated again and excreted. So in the typical metabolism of isoniazid, it's acetylation once, acetylation twice, and goodbye. If you're a poor acetylator, what do you think about the rates of those two events? Is it easy for you to acetylate once? No. How about to acetylate twice? Now it's even harder. Right? So if you're lucky enough to acetylate it once, it's going to be hard to acetylate it a second time because you're a poor metabolite. So along comes our toxic P450. Remember how I said this drug can go through phase 2 and then go through phase 1? It can do that in slow acetylated. So in phase 2, it's acetylated, it's hydrolyzed. But because there's such poor efficiency in doing the second reaction, the 2E1 actually does a phase 1 reaction on the compound to make it a severe hepatotoxin. So here, phase 2, spontaneous hydrolysis, phase 1, and hepatotoxicity. No good. Okay? And so the reality is it would be great to know, it would be great to get isoniazid into 2 billion patients that are latently infected, get rid of this disease forever. But you know that that'll cause drug resistance, people won't take the drug as prescribed, you got to take it every day for 8 months to a year, all those issues. And then how are you going to screen for liver toxicity in a lot of these places where they're not equipped? Right? So anyway, just some thoughts on how this becomes relevant. 2D6. 2D6 is an interesting one. They're all interesting. Everything's interesting. So here we go. Here's a drug called dibrisequin. It used to be used for hypertension. And this was the original, one of the original studies published on it that showed not only the presence of wild-type extensive metabolizers, not only the presence of 4 metabolizers, right here, look at the ratio, like 100 times higher than here, 4 metabolizers. But here was the identification of some ultra-rapid metabolizers. So in your article, if you're as inclined to do it, if you may be inclined to do it, if you read the intro section of the article, it talks about this whole history of how we started to understand from a phenotype approach. Again, this is phenotype approach. We don't know anything about the genes. And we just know about the metabolites that are measured in the urine or in the blood. I can't remember. Yeah, here's urine. So 2d6 is a bizarre one because not only can you have a good copy or a bad copy, this one can skip. You can have multiple copies of a good copy. Multiple copies of a good copy. So how does that look? So there have been patients identified who have up to 13 functional copies. So somewhere in their lineage, the DNA replication went berserk. It got stuck, and they copied the gene 13 times. Duplicated 13 times before it reset itself. So here are some people that have 13 copies of 2d6. They're called ultra-rapid metabolizers. Is that very common? No. But it's been observed in the human genome. So here we have a situation. These squares that I'm pointing to would be a person who has no 2d6, completely deficient in activity. So you give them nortriptyline, which is an antidepressant, metabolized by 2d6, and it stays in their body for a long time because they have no 2d6. So there must be some other P450 that metabolizes it. But compare that now to the person who has 13 functional copies. It's resonating long enough in the body to have efficacy. Right? They get rid of it so quickly, and then you have these shaky brain. More number of functional copies, more quickly the drug is released. And so that's a weird example. You can have multiple copies of gene duplication mutation. And so if we were to look at our situation here, kind of try to sum it up in this patient. This is a patient who had a lot of problems. So first, let's just say they had a lot of problems. The patient had renal failure. The patient was being treated with systemic antibiotics that inhibit cytochrome P450-3A4. And the patient was given codeine in the emergency room. Now, codeine is generally considered a mild opioid. And the reason why it's generally considered that is it's usually metabolized by CYP3A4 and glucuronidated and eliminated. But in this patient, there was a bizarre kind of issue because their antibiotic was inhibiting the 3A4. So could the codeine be metabolized this way? No. So a second P450 that's able to metabolize codeine is 2d6. And it's usually the minor pathway. Usually it metabolizes some of the codeine to morphine. Morphine gets conjugated. And morphine was a weird one. Remember, it's an example of a drug that retains activity despite being phase 2 conjugated. And so morphine glucuronides are active. And they're even absorbable into the brain. They're as active or more active than the parent compound. So here's a situation where you usually make a little bit of morphine, but eventually it will get excreted. But can it get excreted? The person's in renal failure, so no. So, all right, a little bit of morphine maybe wouldn't have caused the severe respiratory depression that we saw. But what happens in this patient? This patient converted it all to morphine, which stunned the doctors. How could this be? It's not supposed to go down the morphine pathway. And so a lot of morphine was generated because the patient later was discovered to have 13 copies of cytochrome P450-2d6. It was a rare polymorphism, a rare example of a patient with gene duplication. So all of it went to morphine. Morphine got glucuronidated. The glucuronide form of morphine is active as a opioid and cause severe respiratory depression. And probably this wouldn't have been as
interesting as it was, if there could still be some excretion through the kidney, then the kidney was failing. Right, so the doctors who worked on this case identified ultra-rapid CYP2D6 metabolism. Right, and this was one of the first studies to talk about ultra-rapid in the clinic and having a clinical consequence. It was published in a journal called the New England Journal of Medicine, which is one of the premier medical journals in the country. Right, so if you publish in the New England Journal, that usually means that it's a pretty important observation. So this is an example of kind of, why are we using it here? It kind of brings together pharmacogenetics and gives it a pharmacogenomics flavor. Because here we have each single gene, right? Now we're talking about the whole patient, and there's a lot going on. 3A4 is blocked. Right? The kidney is impaired. Morphine glucuronide, weird example of a phase 2 conjugate with activity, and it was like the perfect storm to get this kind of weird response. So hopefully you found that somewhat interesting.